Pratyush, Narayan. I'm HCS Class of 2006, and I attend University. My project was on micronucleus formation in total body irradiation study samples. I worked in Columbia University Medical Center, and my mentor was Dr. So some background. What is total body irradiation? Well, when the patient has a disease such as leukemia, lymphoma, aplastic, and multiple myeloma, they're given radiation to their whole body, which is called total body radiation. It's given through a megavoltage photon beam and into the bone marrow of the person. And it's given in conjunction with chemotherapy, because cancer now, especially now, is treated not just in one manner, but they use chemo, they use surgery, they use radiation to make it more effective. Micronucleus assay. This is an assay which is basically a cell line where what we did was we used a chemical to stop cytokinesis, which, as you all know, when the cell divides, it goes into anaphase. But right before anaphase, the nuclear membrane itself divides. And before the cell can divide, there's two nuclear membranes. We stop the cell at that point from dividing. And I'll show you why that's important. HBBLs are human peripheral blood lymphocytes, which are white blood cells. And we use those because they have one of the larger nuclear membranes in the human body and less cytoplasm compared to other cells. Micronucleus formation. So as you all know, radiation is bad for you. Why? Because radiation is ionizing. It forms ions, which if you've taken chemistry, ions are charged particles, and they can damage the cell and the nuclear membrane when the cell is dividing. And what can happen is when the chromosomes are dividing, they might get deleted, it might break away, like a part of the chromosome might break off and go outside the nuclear membrane. Now the cell's going to say, well, that's a chromosome, so there should be a nuclear membrane around it. As you can see, these are the two membranes, these are the two nuclear membranes, and when the cell divides, both have one nuclear membrane each. But this is a micronuclei, so there must have been a chromosome or a fragment or anything that could have broken off, and the cell thought, well, as part of the nucleus, form the nuclear membrane. Now when this divides, one of the daughter cells is going to have more than one nucleus. That's abnormal. So that's the way we can use micronuclei as a DNA damage marker, as a way to find out how much damage has been done. Grade is the unit in which we measure the dosage of radiation given. 1 grade in chemistry terms will be 1 joule of ionizing radiation by 1 kilograms of matter. In simple terms, it's basically energy. It's 1 joule of energy. And the machine that we used to give radiation to our cells was emitted 0.86 gray of gamma ray per minute. So our objective was to determine the effect of radiation-induced DNA damage in HPBLs by counting micronuclei. Because what happens is when patients are given radiation, the machines can make mistakes. So Memorial Sloan Kettering, which is one of the prominent cancer centers in the country, they send us blood samples. And what we did at first was, our objective was a normal healthy patient who does not have a condition that affects their cells in the way that micronuclei will be formed. We took a normal healthy patient's donor and we took their blood. And then we gave it radiation from 0 to 8 grade. So we grew it in vitro, which is basically in incubation outside the body, and in 8 different flasks. And we gave each of them radiation from no radiation to 8 grade of radiation. And we counted the micronuclei in it. And when we counted the micronuclei, we just set up a curve. Okay, there's supposed to be about this percentage of micronuclei if there's, let's say, 5 grade of radiation. So then when we get the patient sample and we harvest it on a slide, and we count, okay, there's 30% here, we could tell, okay, this person received 3 or 4 grade of radiation. That's important because One of the interesting things that I found was, I don't know who's in like college right now, but a lot of college universities, in front of their dorm buildings, they have like newspaper stands. And one thing that caught my eye one day, I was walking back to my dorm, and there was a New York Times article, and it said, as technology surges, radiation safeguards lives. And it talked about how a patient received 38 overdoses of radiation. And because of that, right now he's in a wheelchair with a feeding tube, because he can't eat anymore. That's how badly he was damaged. And this was because the hospital received a very new machine, but even with the instruction booklet, the hospital staff made a mistake. And so the person kept getting over-radiated. The materials we use, the lymphocytes, as I already mentioned. PHA, which is a chemical that simulates cellular division, so that we can split cells into different flasks. We use the medium to grow the cells in, which provides nutrition. Cytoplasm B, is used to prevent the cytokinesis. This is used to stop the cell before it can divide. And then we use fixative to fix it on the slide and make sure it doesn't decay. And DAPI was the stain we use, which is a fluorescent stain, which I'll show you why it's important. Some of the equipment were light and fluorescent microscopes, flasks, tubes, centrifuge tubes, the gamma ray chamber where we gave the radiation, pipettes, slides, and most of the other things, and the computer, of course. So the procedures, we collected the blood from a patient, we irradiated them, we added PHA to it in the culture, we waited 44 hours, added the chemical to inhibit the cellular division, then we used KCl to harvest it, and then we put it on the slide. And then we used DAPI and vector shield to mount it on the slide, and then we were able to see it. So now, this is why micronuclei is important. Because when you're assessing damage, you can do it by assessing chromosomal damage, which I did my first day in the lab. And what you get is pictures like this. But then since it's a fluorescent microscope, as you know, a chromosome is double-stranded. We break the strand and we add a synthetic probe, which is basically a synthetically made DNA strand. And it goes and attaches to the DNA. And then it has it fluoresces under the microscope when you emit a light. So that's why you're able to see different colors for different chromosomes. But basically it comes like this on the slide. You have to cut it out properly, and then the software will put it together, but then when you see the damage, you have to actually assess each of these, and you have to score each of these. Now, each of these studies requires 500 to 1,000 cells. If you do this, you're doing about 5 or 6 a day, so it's going to take very long. Now, I'll show you why micronuclei is important, because you're going to have thousands of this on just one slide, and all you're doing is you're looking under the microscope, going through it, and just counting the micronuclei. So 1, or if there's 2, you put a 2, and so on. So 500 to 1,000 cells can be done very quickly compared to you doing chromosome analysis. And also, one of the things that affects patients that you have to keep in mind is this is a Chinese.
In New York in July. If you were going.
This is the difference between a fluorescent and a light microscope. Pretty much in a light microscope, the light comes with reflect and you're seeing it straight. Whereas a fluorescent microscope is emitted through a machine and through a filter. And then when it hits the chromosome, the light that's excited, it fluoresces, and that's the color you see. And that's how you can do chromosome analysis, because each chromosome has a different emission. The samples we collected for the micronuclei were before the treatment, before they were given radiation, right before it, 4 to 6 hours after. So we got blood from them 4 to 6 hours after, and then 24 hours after, to see how the micronuclei has changed.